Welcome to the Biological Life Sciences students. Is my screen visible to you, all of you? And the voice is visible to you? Please reply. Is the screen clear to all of you students? Okay. So since voice and all is clear, we will go through the lecture. We'll start our lecture now. Okay, so the topic for today's lecture is instrumentation course in life sciences. Okay, so let's see what all are the topics that we are going to see in this. What all are the different things that will be covered in this. Okay, so today's topic is from unit number 4, that is SDS page. Okay, so we are going to begin with the SDS page now. So let me first introduce tell you the full form of this SDS. SDS means sodium dodecyl sulfate. Okay, it means sodium dodecyl sulfate, and page means polyacrylamide gel electrophoresis. Okay, page means polyacrylamide gel electrophoresis. Okay, so here there are two different uh, monomers that is used. First of all, first monomer that is used is acrylamide, and the second monomer that is used is bisacrylamide. Okay, so there are two different monomers that is used, acrylamide and bisacrylamide, and SDS is involved here, or SDS is used to give a charge, to give a negative charge to the overall protein. Okay, so therefore we are going to see how this is done in actual. Okay, so you can see here this shows a unit where SDS page is being done. This is a vertical electrophoresis unit where actually the protein samples are being loaded. Okay, you can see here it is shown over shown to you here in this. Okay, so this is the unit where the vertical electrophoresis happens. Okay, so the electrophoretic mobility of the protein means the protein, how it is going to move in the SDS page gel. It depends on the electric field. First is electric field. Okay, then it depends on the molecular weight. Okay, there are two things on which it depends. First is electric field, and second is its molecular weight. Okay, so now let us see the principle that governs the SDS page. Okay, how to prepare a SDS gel, how we can prepare a gel, or how to analyze the gel, or how to stain the gel. Okay, the proteinic bands are not visible by default. Clear? The proteinic bands are not visible by default. It has to be stained using different stains. Two of the stains that we are going to discuss in today's video. First is Comasi brilliant blue stain, and the second is silver staining. Okay, so these two things we are going to see. Okay, so now the first important thing, remember, protein is always having a three-dimensional structure. Okay, so since the protein is having a three-dimensional structure, so how it is going to move into the agarose gel? Okay, so that we are going to see. So first of all, the protein structure, that is tertiary, secondary, or quaternary structure, has to be converted into a primary structure. Okay, it has to be converted into a primary structure. Primary structure of protein means what? Primary structure of the protein means a linear structure, or the linear protein. Okay, this will not have any kind of three-dimensional structure. It will just have a polypeptide chain. Okay, so in order to move these protein bands in the agarose, sodium dodecyl sulfate page gel, you have to convert this into a linear structure, and this is done by heating or boiling the sample. Okay, the sample is boiled into the water bath, and it is converted into a straight linear structure like this. Okay, so now let's see how the gel is formed, or what are the different monomers that forms the gel. Okay, you can see here first monomer, that is NN methylene bisacrylamide. This is a cross-linking monomer. This is a cross-linking monomer which actually helps in polymerization. And the horizontal linking monomer is acrylamide. You can see this is the structure of acrylamide monomer, and this is the structure of cross-linking monomer. Okay, they combines together like this. Okay, this bisacrylamide, it forms a vertical bond like this, and the acrylamide forms a horizontal bond like this. Okay, so after forming the two bonds, it forms different pores. Okay, you can see it is properly shown to you here in this. Okay, you can see it is shown to you here in this. This is the cross linking, vertical cross linking between the vertical cross linking by bisacrylamide, and this is the horizontal cross linking by acrylamide. Okay. Okay, is it clear to all of you? Okay. Now let me show you once again the structure of bisacrylamide. See, this is the structure of bisacrylamide, and this is the structure of acrylamide monomer. Okay, acrylamide monomer is going to form a horizontal linking, whereas bisacrylamide monomer is going to form the vertical linking. Okay, it is going to form the linking like this. Okay, the linking will not happen or start unless there is a timid, or unless there is a free radical that is given in the reaction mixture. Okay, this free radical is generally the ammonium persulfate that we add. Okay, we add the ammonium persulfate and then we add timid. Timid is used for catalysis. Actually, timid fastens up the reaction, that is the free radical catalysis, or the free radical that is provided by ammonium persulfate. It helps us in polymerizing the gel. Okay, timid makes this process faster, so therefore both of them is added into the this gel mix in order to polymerize the gel. Okay, so you can see there are two different types of gels. Okay, as Page is always having two different types of gels. First gel is stacking gel, and the second gel is running gel or resolving gel. Okay, stacking gel generally it is having 0.125 molar of tris HCl with pH 6.8. Okay, generally the acrylamide strength is 5% here. It has a larger four pores and lower ionic strength, so therefore the lower pH also. The running gel or the resolving gel is having higher concentration of acrylamide, means the pore size will be smaller. Therefore, it helps in resolving or separating the different protonic bands. And the molar strength is 0.375 molar tris HCl. And the pH of tris HCl is adjusted to 8.8. Okay. So this can be adjusted according to the protein size. Okay, if you are doing the electrophoresis of different kinds of protein where you require a larger pore size or a smaller pore size if you require, then the size of the gel can be, or the pore size of the gel can be reduced or increased depending on the acrylamide concentration present in the gel. Okay, is it clear till now? Is it clear to all of you? Okay, so let's go further. Okay, now you have understood that there are two different gels. First gel which comes on the top, which is having the wells like this, is called stacking gel, and the gel which comes below into the plate is also called as resolving gel. Okay, so let's see one by one how this happens. Okay, so now there is a key component in the proteinic separation, that is the ionization of glycine. Okay, there are different amino acids that is present in proteins. Now here the gel buffer or the running buffer that is used, or in the SDS page, is kept in a buffer. Okay, and this particular buffer is generally made up of glycine, so it is called as glycine SDS gel running buffer. So in common terms it is also called as reservoir buffer. Okay, so the glycine which is present into the gel running buffer, or the major component of gel running buffer, is having a zwitter ionic form, where it is having neutral charge. Okay, zwitter ionic form of amino acid means it is having a neutral charge. Total charge is neutral. Okay, negative as well as positive charge is available, and the proteinic molecule may be having a positive charge or negative charge. Protein can be positively charged or negatively charged. Yes. So in order to move the protein in a standard positive direction, it has to be coated with SDS. And therefore, in this page, that is polyacrylamide gel electrophoresis system, we add sodium dodecyl sulfate, that is an anionic detergent, and it provides the positive charge. Yes, it provides the positive charge to the proteinic molecule. Okay, and after providing it provides the negative charge to the proteinic molecule, and after providing the negative charge, the protein is ready to move in the positive side. Okay, so you can see how the protein will be moving in the gel. The relative pro of the protein depends on the distance traveled and the different also. Okay, so more the molecular weight of the protein, it will be lagging behind less. It will be moving very slow, or less the molecular weight of the protein, it will be moving very fast. Okay, so you can see it is shown over here where the smaller molecular weight proteins are moving forward and the larger molecular weight proteins are lagging behind. Okay. So this is how the buffer is poured onto the electrophoretic tank. I am going to show you the electrophoretic tank also. The proteinic bands get separated on the basis of the pH. Okay. The first SDS gel running buffer is having the first gel, that is stacking gel, is having a pH of 6.8, and the resolving gel is having a pH of 8.8. Okay. So there are two things onto which the protein bands get separated. First is the pH difference in the gel, and second is the difference in the pore size or the gel acrylamide concentration. Okay, so what is the role of stacking gel here? Whenever you load the protein bands or proteins in the well of the SDS gel, these proteins get stacked onto the stacking gel. Okay, and once it enters into the separating gel, it starts separating out. Okay, so you can see we are loading the sample here onto the wells that is made in the stacking gel. These wells has to be made using a comb, and then the sample is loaded, and after the sample is loaded, the electrophoresis is start. Okay, generally till the sample the sample protein sample that you have loaded comes into the gel, you have to put the electrophoresis at 15 voltage, and after that you can increase the voltage to the 100 voltage. Okay, so you can see protein stacked up in the stacking gel. All the proteins will stacked up here, and when it reaches the interface of the separating gel, it will start separating. Okay, you can see the migration of protein is invisible. Why migration of protein is invisible? Because protein is now not stained here till now. Okay, so you cannot see the proteins, so you only see the blue colored sample loading dye. Now this sample loading dye is having a bromophenol blue in it, which helps in tracking the protein. Then it is having glycerol, plus it is having glycerol or sucrose. It is having beta mercaptolithanol also. What is the role of beta mercaptolithanol in the sample loading dye? It makes the protein, or it reduces the disulfide bridges that is present in the protein. Okay, disulfide, it breaks the disulfide bond, and it it keeps the protein in a linear form. Okay, it does not allow it to form a secondary or a tertiary structure. Otherwise, it will not move in the gel. Okay, so now how this gel is analyzed, you can see different solutions are first prepared, that is stacking gel buffer, then separating gel buffer. Okay, then ammonium persulfate timid is there. Okay, if you are not having the ammonia, see, I'll tell you one more alternative way. If you are not having APS and timid, then also you can polymerize the gel. Okay, how you can do it, I'll tell you. Okay, you can see there are different plates. First plate is notched one, and second plate is plain one. Okay, so that you can put up the comb to that plate. Okay, in this case the upper plate is more larger, and the smaller below plate is smaller. Okay, these plates are very fragile as these are made up of glass. Okay, this has to be very much clean before performing the electrophoresis. Okay, so you can see the type of sliding clamp that is given to you. Using this clamp, you have to attach the uh, two different plates, and in between which you are going to cast the gel. Okay, this casting gel assembly comes with the electrophoresis unit that can be of different different companies, and all they produces this kind of units. So you can see these plates are sealed from both sides and below also, and then it is ready to pour the buffer. First of all, it has to be tested for leaks with deionized or distilled water. If there is no leak, then you can go forward. You have to mix start the mixture, making mixture of the separating gel first, because first of all you have to pour the separating gel into the agarose gel electrophoresis assembly plates. Then you have to put the stacking gel mix. Okay, So you can see here, first of all, what we have done here, we have assembled the gel into a test tube or a beaker. Okay, then you pour that gel into the plate, gel between the plates. Okay, the resolving gel concentration may be of different types. See, depending on the protein concentration, the resolving gel may be of different type. Okay, so you can see if you are preparing the 8% gel, then you have to take 4.6 ml water and 30 ml acrylamide solution. You have to take 2.6 ml, and then trees HCl pH 8.8 you have to take 2.6 ml, and then 10% SDS 100 microliter and 10% APS 100 microliter, and timid you have to take 10 microliter. Okay, so likewise, the concentrations of different gel for the resolving buffer, for making the resolving buffer, is shown to you here. What you can do, you can take a screenshot of this page so that uh, you require all the concentrations will be available to you as and when you require. Suppose if you want to prepare a 15% resolving gel, then you have to add these all these components in the desired concentration that is shown to you over the top of this table. Okay, but remember, timid and APS has to be added at last. Okay, APS, provides a free radical that is used for polymerization of acrylamide and bisacrylamide. Timid makes this process faster. Okay, so remember, as soon as you add 10% APS and timid, the gel starts polymerizing. So you have to immediately pour that gel onto the uh, onto the plates, okay, in between the plates immediately. Okay, and while pouring the gel, you have to ensure that there is no air bubble trapped, because the air bubble will have O2, and O2 stops the polymerization process, or it limits the polymerization process. Okay, so this was the concentration of dissolving gel that was shown to you here. Now let's see the next gel, that is stacking gel. Okay, generally the stacking gel is made 5% because its role is only to stack the proteins one upon another, and when it reaches this separating gel, it starts separating out. Okay, so 5% gel is used here. The concentration of 5% gel, stacking gel, is shown to you here. 5.86 ml, take water, then 30% acrylamide, you have to take 1.34 ml, and then you have to take 0.5 molar tris HCl, that is pH 6.8 solution 2.6 ml, 10% SDS solution 100 microliter, and then APS 10% 100 microliter, and 10 microliter timid. Okay, so this will give you a concentration, or this will give you a gel of about 10 ml. Okay, 8 to 10 ml gel you will get from here. So you can change the concentration as and when you require the total amount. Okay, so depending on the plate size, if your plate size is smaller, you can use that much gel and adjust the concentrations here. If the plate size is bigger, you can increase the concentration as per the given table. Okay, if the polymerization is not happening by APS and timid, okay, generally it will happen. But if it is not happening, or if you are not having a solution such as timid and APS, then you can go for alternative way that is called as photopolymerization. Okay, so now let's see what is a photopolymerization. Okay, so in the photopolymerization, you can in the photopolymerization, what you can do, you can use riboflavin. Okay, so how this riboflavin helps in photopolymerization, that we are going to see. Okay, just a minute. Okay, so what you do, you just add riboflavin in the acrylamide and bisacrylamide mixture. Okay, what you have to do here is you have to add a high light source. Okay, you have to use a light source here. Okay, so you can see here, ammonium, instead of ammonium persulfate and timid, it is replaced by riboflavin. Okay, and when the gel is poured, it has to be poured in front of a bright light for 2 to 3 hours. Okay, you have to keep the gel plate in front of bright light, specifically sunlight, if it is better, for 2 to 3 hours. Okay, so what is going to happen, this helps the riboflavin to photopolymerize and generate free radical. Now this free radical initiates the polymerizing process. Okay, so you can see acrylamide plus bisacrylamide in the presence of riboflavin under light for 2 to 3 hours gives you the page gel, that is polyacrylamide gel. You have gel is prepared. Okay, so this is an alternative way how which you means how you can prepare a gel. Okay, so after electrophoresis, the procedure that is followed will be, carefully you have to remove the gel from the tray, and then you have to add a gel fixing solution, and after gel fixing solution you have to remove it, and you have to add Comasi brilliant blue solution. Okay, so it's a generally blue colored dye, and you have to keep it on a platform rocker, a rocking table, where the strain will be evenly spreaded. Okay, you have to stain the gel for 2 to 4 hours if you are using Comasi brilliant blue, and you have to use the de-staining solution to decant the excessive stain. Okay, after 2 to 3 hours or 4 hours, it depends on the concentration of Comasi brilliant blue also. If it is very dilute, then you have to keep the gel overnight, and if it is very concentrated, you can keep it for 2 to 4 hours. Okay, remember, when you keep it for 2 to 4 hours, the protein bands will be stained along with the whole gel. So you have to go for de-staining step, where you use methanol and glacial acetic acid solution, and you have to de-stain the gel. Excessive Comasi brilliant blue stain is removed, and only the clear protein bands will be seen. You can see the only clear protein bands. Okay, so I will show you how the gel looks like after the protein bands are. See, this is how the protein bands are seen. Okay, you see, this is the whole process where you have to, add the gel into the plates. Okay, then you have to check it with distilled water, and then you have to add the comb also. You can see here, it is shown to you here how the comb is added on the top. Okay, you can see this interface is actually what it is, the interface or the difference between the stacking gel and the separating gel. The upper one is the stacking gel, and the lower one is the separating gel. Okay, and you put the comb, and slowly you remove the comb afterwards, after the gel is polymerized. Comb has to be removed very much slowly. Why? Because that it may distort the wells. Okay, so you have to add distilled water here, so that the vacuum that is produced is nullifying the effect, and the empty space is taken up by the distilled water. Okay, then later you can decant the distilled water. Okay, so this is how the this is done. Okay, you can see how the assembly is placed into the casting tray. Generally, advanced electrophoretic system has this casting assembly, but a regular electrophoresis assembly that is generally used in academic institutes and all will not have a casting assembly like this. There you have to manually put the plates between the Teflon spacers, and you have to pour the gel by locking the gel and not allowing it to seep out. You have to put the gel assembly into the tank. Then you use a hairy root tip like this to load the sample. You can see how carefully the sample is loaded here, and you have to put on the electrophoresis. Okay, this is how you put on the electrophoresis. So this is how the gel looks like. Okay, once the electrophoresis is over, this is how the gel looks like after running. So in certain kinds of DNA ladders or protein ladders are pre-stained. Okay, these protein ladders need not to be stained separately. Okay, you can see here, you can see this orange colored protein ladder here. This is already stained. Okay, but for protein bands you have to stain it separately. Okay, you have to do it separately completely. Okay, so this is how the gel is going to look like. Okay, see here, this is the gel and the blue colored band. This is a de-stained gel where the proteinic bands are seen specifically, and here the ladder is added. Okay, so this is the way by which you can do the this Comasi brilliant blue staining. Okay, now we are going to see this is the structure of Comasi brilliant blue. By the way, you have to stain the gel in a staining tank like this. Okay, Comasi brilliant blue G250, its name is. You can see the structure of the Comasi brilliant blue here. Now the silver staining here, you have to use a silver nitrate solution. Generally, silver is having an affinity towards the protein, and it forms a dark brown band. Okay, so I'll explain you the procedure for silver staining. First of all, rinse the gel with the ultra pure water, then fix the gel using fixing solution, which is generally acetic acid and ethanol. Okay, this will remove all the different compounds that are not required in the staining. Then you have to add 30% solution and sensitize the gel using silver sensitizing solution, that is 10 ml. Okay, then you have to wash the gel again with 30% ethanol. Again, wash the gel with water. Add the staining solution, that is generally silver ion containing solution. Generally, silver nitrate solution is used here. And after staining, rinse it briefly with the ultra pure water, that is distilled water. Incubate it for 4 to 8 minutes, and then excessive silver staining solution is removed, and then again wash it with distilled water. Okay, so this technique, that is silver staining, does the staining quickly, means it requires low time. Okay, so this is how the gel, that is SDS page gel, looks like when you do a silver staining. Okay, there is a brown colored band that is seen. Okay, you can use it for determining the molecular weight also. Okay, you use a DNA ladder of the known molecular weight, and then unknown samples molecular size can be determined using a graphical method, just like we do in estimation of different components. Okay, so thank you so much, students, for joining. This was all about the STS page that I wanted to share with you. I hope the whole thing is clear to you. If any doubt, you can comment any time in the comment section. I am ready to give you answers to your questions. Thank you for joining, all of you. Thank you so much.